In this video, we want to cover diphtheria lab diagnosis, okay? So, assuming that you know what diphtheria is, everything, now you want to write the lab diagnosis. This is the only thing they have asked you, lab diagnosis of diphtheria, okay? So, the first thing you will collect is a specimen. Specimen collection is the first step you have to write. You will collect throat swab. See here, you will collect a throat swab and a portion of membrane, pseudomembrane covering the tonsils. You can see here, bull neck appearance, pseudomembrane covering the tonsils, classically seen in diphtheria. So, the specimen will be a throat swab and a portion of the membrane. This is cutaneous diphtheria. They have not written it in the lab diagnosis specimen. Okay, now coming to direct smear. This is the next step. Second step is what? Direct smear. Now, whatever you have collected, you are going to smear it. This is going to be gram stain. The center diagram is the gram stain. Club shaped, gram positive. It's a gram positive. You can see purple, right? You can see purple color in the picture, the middle one. So, it means what it is? Gram positive. You look at this one. Gram positive. I hope you are looking at the right diagram. This one, guys. So, it is a gram positive club shaped bacilli with Chinese letter arrangement. Draw this diagram in exam, you will get marks. Okay. Then this is Albert stain, the third one. This one is Albert stain. Oopsie, 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 oopsie. This one is Albert stain. So, Albert stain, green bacilli with the bluish black metachromatic granules. Green with bluish black metachromatic granules. Let me at least put the shape of the how you should draw the green bacilli. Got it right? A green bacilli you should draw here. At the end there will be a bluish black metachromatic granules. You will arrange them in Chinese letter pattern. And diphtheria in gram stain you will draw a purple line. Okay? Very good. Shall we move on? So, in diphtheria lab diagnosis you will draw a gram stain diagram and Albert stain diagram. Very good. Coming to culture media. Now, you will also put it for culture. You will put it on culture for, on Loeffler serum slope, you will put it for culture. Okay, put it on culture for, on Loeffler serum slope and also put it on potassium tellurite. These are important ones, okay. You should not forget potassium tellurite and Loeffler serum slope for Corvinibacterium diphtheriae. Black colonies on potassium tellurite. On potassium tellurite, it will be black colonies, remember. Biochemical identification, urease will be negative just like E. coli. Urease will be negative. His serum sugar media, it ferments glucose, maltose or starch. His serum sugar media, we'll come to the details, okay, just look at these for now. His serum sugar media, biochemical identification for diphtheria. Then moving on to diphtheria toxin demonstration. Please wake up if you are sleeping. Take a deep breath. Okay, now we have come to the 5th slide, is it? How many slides did we look at till now? Specimen, direct smear, culture media, biochemical identification. Last one, demonstration of diphtheria toxin. So, we are at the 5th step, 5th point. In this, you have two things, in vivo test and in vitro test. In vivo means obviously it will be animal inoculation, right? So, it will be a guinea pig inoculation. Guinea pig is actually a rodent, not a pig. And it is having subcutaneous and intracutaneous tests you can do, okay? In vitro test, you have Elex gel precipitation test, detection of tox gene by PCR, detection of tox gene by PCR. Then you have detection of toxin by ELISA or ICT. ICT is nothing but immunochromatographic test. Then you have cytotoxicity on cell lines. First, let us try to revise this much. Then we will go into the details. Okay, revision. What and all are there? 5 steps are there, correct? Specimen. Specimen, what you will collect? Specimen. Specimen, you will collect throat swab, portion of membrane, correct? Then you have direct microscopy you will do gram stain, Albert stain. Gram stain, Albert stain. Then you have culture. Culture, very specific. You should write Loeffler serum slope. Loeffler. You should learn the spelling. Loeffler serum slope and potassium tellurite. You have to draw the diagrams of all these. Potassium tellurite, Loeffler serum slope, Albert stain, gram stain. You should draw the diagrams. Then what was the fourth step? Fourth step was biochemical investigations. There we saw some His sugar serum something, right? And then we saw urease is negative. Then the last slide we saw diphtheria toxin. Diphtheria toxin determination. Diphtheria toxin determination, you have what and all you have in diphtheria toxin determination, in vivo and in vitro. In vivo you will do animal inoculation for guinea pig, etc. In vitro you have Elix gel precipitation test, ICT something, immunochromatographic test or something, and then you have tox gene you will find out, and cytotoxicity. Okay, so this much we have done for now. Let us meet in the next video for more.